Hi, my name's Clay Isom. I'm an associate professor in the Department of Animal, Dairy, and Veterinary Sciences at Utah State University, and I'm really happy to be able to talk to you a little bit about the grazing research that we've initiated. This project started out looking at beef steers and grazing a variety of different forage types, forage combinations, grass only, fertilized grasses, grasses in combination with different types of legumes. The research started looking at tall fescue in combination with bird's foot trefoil, which is a non-bloating legume, or alfalfa, which is more typical of what we see here in the Intermountain West. The results of that project told us that the greatest average daily gains in beef steers came when tall fescue was planted in combination with bird's foot trefoil, which went a little bit contrary to our original starting hypothesis, which was that alfalfa, grazing alfalfa with a bloat protector block would result in the best gains. We realized that there wasn't a lot of research being done on forage being fed to organic or grass-fed dairy animals here in the Intermountain West. And we thought that it made a nice logical progression to look at these different forage combinations in developing dairy heifers, specifically at this point, in follow-up to our previous study with the beef steers. One of the additional findings that came out of the previous study with the beef steers and the different forage combinations was that the combinations of tall fescue and bird's foot trefoil or alfalfa didn't yield the type of intakes and gains that might have been expected otherwise or predicted by various formulas that are out there in the scientific literature. And so we devised an experiment that was a combination of 4 different grasses, which included tall fescue, meadow brome grass, perennial ryegrass, and orchard grass, a high sugar variety of orchard grass, planted either alone or in combination with bird's foot trefoil. And we planted these in a series of plots at our research facility in Lewiston, Utah. And on these grazing plots, we turned out a number of young prepubertal or peripubertal Jersey heifers. And the aim of this experiment was to see how these developing Jersey heifers would respond to the grazing experience, and specifically how they would respond to the different forage combinations, either the different grasses alone in monoculture or in combination with the bird's foot trefoil. There's some suggestion in the literature that in highly fertilized pasture, the blood urea nitrogen content in growing heifers or even lactating or growing dairy or beef cattle can reach highly detrimental levels, specifically to reproductive performance. A lot of interesting data coming out of this. We've got a multidisciplinary team, very broad and talented team from lots of different departments here at Utah State University. We're involved with extension personnel all across the Intermountain West. And each investigator or each part of this team is finding very interesting and valuable information in their own right. We're looking at a variety of other endpoints, growth, nutritional physiology, rumen health and well-being of these animals, behavioral standpoints or endpoints such as rumination and reproductive behavior, as I mentioned. We're looking at a variety of different environmental endpoints that would help us to understand how nitrogen and other nutrients, phosphorus for example, are cycling through the environment in these different pastures, either in monoculture pastures or these mixed pastures, and how that might relate to concepts of environmental sustainability. We're also undertaking an economic analysis of these endpoints that take into account such factors as the yield, the production characteristics, the dry matter intake, the growth of the heifers, their performance.
Hello, I'm Rhonda Miller, and we're at the Utah State University Intermountain Irrigated Pasture Facility at Lewiston, Utah. Today we're going to look at a study that's examining nutrient cycling in an organic grazing system. Specifically, we're looking at different forages, grasses, with and without bird's foot trefoil, a legume that contains tannins. And we're looking at how those nutrients impact the rate of gain, the nutrient cycling, and especially how much is being lost in the leachate, and also how those nutrients impact the reproductive growth of our developing heifers. If you'll come with me, we'll go take a look. We're here on my plots. Ours are on some skinny strips so that we can cover or match some zero tension lysimeters that were previously installed in this field. We're looking at the nutrient cycling or environmental impact component of this study. And so you might be wondering why we're looking at nutrients in a grazing system. You might think a grazing system is perfectly benign. In many ways it is. I mean, you're pulling the nutrients from the grass, and all those nutrients for the most part are being recycled back onto that pasture. But when you actually stop to look at it, those nutrients are not being recycled uniformly. Within any given year, only about 10% of the pasture receives a urine or fecal spot. 90% of that pasture isn't receiving any nutrients back on that system. Those nutrients also tend to be highly concentrated around the water trough. And when we actually look at a urine spot, those nutrients in that urine spot are the equivalent of about 1,000 pounds of nitrogen per acre. Now, only in that urine spot, the rest doesn't have that. But 1,000 pounds of nitrogen per acre is way too much for any of our plants to utilize. The urine is also primarily made of urea, which rapidly converts to ammonia and then is volatilized, which creates its own environmental issues with acid rain, and especially in the case of northern Utah, some PM 2.5 issues. The nutrients that don't volatilize and that the plants don't utilize or intercept, those move down and get into our groundwater system. We don't want nitrates in our groundwater. If we get too many nitrates in our water, it can actually result in blue baby syndrome, and in certain cases that can actually result in fatalities. It's also associated with some spontaneous abortions in livestock. So we're trying to find forages that utilize those nutrients just as efficiently as possible and minimize any nutrient loss, especially through leaching. Okay, and here we are at our trailer. This is pretty well the heart of our lysimeters system. The zero tension lysimeters are 15-inch diameter by 44-inch deep cores, soil cores, that we pulled out, and then we put on a reducer and cap system with PEX tubing underneath. Those cores went back into the ground where they were. We have PEX tubing running from various locations in the field all the way up to this trailer, and you'll see we have these little access ports up on the wall. We then put a vacuum onto those ports and pull out the leachate that was collected in the reservoir, and we collect them in these big bottles here, and then we take that back and analyze it for the nitrates and phosphorus that are being leached in the system. So the beauty of the zero tension lysimeters, were installed in the field many years ago. We can actually plant and till over them, so we don't have to disturb them, and they collect all of the leachate through the whole year. So even in the winter when we can't get out here, the leachate is moving down past that soil root zone into the reservoir, and then as soon as we can get out here in the spring, we collect that leachate. But that way we collect all of the leachate through the whole year. What we do see is we see huge flushes of nutrients being moved through that soil profile with the snow melt. And we do get a fair amount of snow here, so that's always a big issue each year. So that's one of the things we really want to look at. Here we are at one of my suction cup lysimeters. We have these in every paddock in this pasture. The suction cup lysimeters are a PVC tube with a little ceramic cap down at the bottom. Those tubes are about 3 foot deep. And we actually have them covered with a metal plate. Inside, an irrigation cover. And then In there is a rubber tube access port. We pull that little ring off, put a little tiny tube down the suction cup lysimeter, put a vacuum on it, suck out the leachate. We then put a new vacuum back onto that tube, close it off, and seal it back up for the next week. And that's how we collect our leachate. So these we collect every week during this grazing season, and we go as early in the spring as we can to as late as we can in the fall. But during the winter, we are not able to collect leachate on the suction cup lysimeters. So here we are at our root cages. We're actually looking at how the plants grow and how the roots, how much of a rooting system there is, how deep that goes, because we think that may help explain why some forages are better at utilizing the nutrients than others. So in these cages, and these cages are just to protect our tubes from the heifers, we have acrylic tubes, and they have a little cap, and there's a little black styrofoam tube that goes in that, and that's to help prevent any light from intercepting, because the light down there would change how those roots grow. We then take a circular scanner, drop that down the tube, and take a scan of the roots at 1 foot increments. We go down for 4 feet, and then we take those back to the lab and we stitch those images together, but we trace them. We can measure the root diameter, the root length, and we hope that that will help explain what's going on, or some part of what's going on as to why some forages are better at utilizing nutrients than others.
A couple of different specific research endpoints that we're looking at that we're really excited about is one is to evaluate the rumen health of these animals. We know that the inputs that go into the rumen can have a dramatic influence on the rumen microbe population and the overall rumen performance and rumen health, which is an essential characteristic for growth and performance of these animals. So every 35 days we've been going into these Jersey heifers and pulling rumen fluid from these animals and trying to monitor changes over time as these animals come from a feedlot setting and then get put onto grass, but also at looking at characteristics of just overall rumen health, such as rumen pH and rumen contents. We will ultrasound them to identify their reproductive status, whether they have initiated their reproductive cycles or not. We'll monitor their internal reproductive organs, as well as some of the external indicators of puberty and development, such as the mammary gland, and tie that together, those together with hormone data that we can gather from the blood that we pull from these animals, and as well as reproductive behavior data that we can pull from the pedometers and some of the biometric endpoints that we're getting from. And in fact, at the end of each of the research years, as we bred those animals, we saw very little suggestion, in fact, no suggestion, that the higher protein content of our mixed pastures were doing anything to compromise the reproductive performance of these animals. This has been, as I mentioned, a 3-year project so far that we have been involved in. We also have a variety of local and regional producers that are involved, stakeholders that provide a wealth of information and suggestions to us as we meet with them on a regular basis to discuss this project. One of the suggestions, or one of the questions that almost always comes up is, well, how will my Holsteins, for example, or any other breeds of cattle, how will they respond in the same way as these Jerseys? We know that there are breed characteristics that set these breeds apart from each other, both in their growth, performance, their response to forage, and different dietary considerations. And so that's something probably that we'll undertake starting this next year, and perhaps moving forward, if we could attract some funding for this project. would be to look at some of the same endpoints, reproductive performance, nutrient cycling, growth performance of the animals, yields of the pasture, dry matter intake, a variety of different parameters that would help us to understand whether different breeds of cattle perform differently in a grazing setting as far as dairy heifer development goes.